Hey everyone, this is Vladimir Jovanovic again, and I'm here to talk to you guys about ANOVA and the different forms of ANOVA. So as a general outline to give you a sense of what I'm going to talk about, because this might be a long lecture, I'm going to cover a bit of background that you need to remember in order to understand ANOVA. Then I'll cover ANOVA, multi-factor ANOVA, some designs that you can do with ANOVA in terms of your experiments, then ANCOVA, multivariate designs, so like MANOVA and MANCOVA, and then basically doing post hoc analysis and what kind of assumptions ANOVA makes and what happens when you violate those assumptions. So first, let's just get a sense of what the purpose of ANOVA is. Basically, ANOVA is when you want to test if there is a significant difference between two or more means. When it's just two means, you might as well just do a t-test, but you can imagine a case where you have three conditions and you want to know is there any significant difference between those three. ANOVA stands for analysis of variance, and by comparing variances we can test for the mean significance. Alright, so let's go into some background things that you should recall or remember in order to understand it. The first thing we should remember is variance. Variance measures basically how far apart numbers are. A variance of 0 will therefore imply that all the values are the same or that they are not in any way different from each other. It's always going to be non-negative, and when you have a small variance, you basically are saying that there's a bunch of data points that are very close together to each other and to a mean, and when it's a large variance, then it means that they're spread out around the mean and from each other. To calculate the variance, you take the summation of each point with the mean subtracted from it and then squared, and then divide the whole thing by n minus 1, where n refers to the number of observations that you have. So the numerator there at the top refers to the sum of squares, or SS for short. We're going to be using that a lot in ANOVA, so just remember SS for sum of squares, and that in particular refers to taking each observation, subtracting the total mean from it, and then squaring that and adding those squares together. In ANOVA, the key feature is partitioning of sum of squares. This allows us to compare the means across different groups and get a sense if they're significantly different from each other or not. In this example, we have a table where we have an experiment that got 6 total observations, and of those 6 total observations, they were split between two groups, group 1 and group 2. Now, within those two groups, we can find the sum of squares for each. So for instance, in group 1, the mean is 2, and the sum of squares is 2 as well. Basically, what you do is you take the first observation and subtract the mean from it, so 2 minus 2 is 0, and square that, 0. Then you take the second observation, 3 minus 2 is 1, squared is 1. And then the third observation is 1 minus 2, which is 1 as well, and you square it again is going to give you 1. So in this case, you had 0 plus 1 plus 1, which is a total of 2 for your sum of squares. Same thing can be done for group 2. We can also take the overall mean, so taking all points regardless of what group they are, and in this case it's 4. But here we can see that the total sum of squares is actually 28 in this case. So the sum of squares within the group is only 2, and when they're added together you get 4. But the sum of squares for the total is actually 28. And so what we can say is that the variance of the within group is much smaller than the overall variance. And this large difference in the group mean allows basically this variance to exist. And to show that difference in variance, we can look here at two figures. So when there's a large between group difference, as we see here, group 1 and group 2, what we can see is that the mean between the two groups will be somewhere around 4, and both of the variances are far outside the bounds of it. In the second example, where we have a small between group difference, even though the mean might be the same or very similar, the fact is the variances overlap so much with each other that there's a pretty good chance that there isn't any significant between group difference. So in order to test this group mean difference, we use the f-test. So as a reminder from 2 lectures ago that I talked to you about, the f-test is great for testing a significant difference in 2 variations. It does this by taking a ratio of the 2 variances and basically testing whether it's significantly greater than 1. F-test is used in the general linear model for model fit. We covered that before. It's also used in multiple regressions and all those other types of linear models. In this case, in that one, you take the mean square of the predictive values and divide it by the mean square of the error. That's actually very similar to the test done for ANOVA, where you take the mean square of the between group variance and divide it by the mean square of the within group variance. The within group variance here is the amount of error that happens within any one group. So, given that we just reviewed those 3 topics, let's actually dive into ANOVA. Now, for ANOVA, we can go back to the example of having 2 groups with differences. We can do a simple t-test to find the difference between them, and that would actually be the easiest. But in ANOVA, you could actually do the same test, and you can use the f-test in order to use the variance to figure out whether it is significantly different from each other. The ANOVA test requires us to figure out what the sum of squares is for the total, what the sum of squares is within the group, and then what is the sum of squares of between the group. So given our old example where we have group 1 and group 2 with those same values as before, we have the sum of squares for each group and the total sum of squares. The sum of squares of within group, you just add up the numbers in the appropriate cells. So in this case, it's going to be 2 and then 2, so the total is going to be 4. Sum of squares for the between group is actually just going to be the difference from the total with the within group subtracted from it. So 28 minus 4 is going to be 24. K here refers to the number of groups, which there are 2 of, and N refers to the total number of observations. These are important for determining our degrees of freedom. In our ANOVA calculation, I actually printed up here a table for you guys so you could do this yourselves. SSB refers to the between group differences, SSW refers to the within group differences. We can see here how the degrees of freedom is calculated, where the between group differences is just k minus 1, so in our case k is 2, so the df here is 1. In the within group differences, it's going to be 6 minus 2, which is 4. And then from those numbers, we can actually get the mean squared between and the mean squared within, or the mean squared error. And then from that we can get a ratio. In this case our f ratio is 24. Now that we have the f value, we can actually calculate the p value, or the significance, by taking in the degrees of freedom of 1 and 4 and inputting the f value of 24. So that gives us a p-value of 0.008, which we can say gives us a significant between group effect. Here effect refers to the between group variances that we calculated, and error refers to the within group variances. We can conclude that the means are significantly different from each other, but we cannot right now from this result conclude which one is larger. All of this can be extended to any amount of groups or subdivisions of data sets, so you can imagine that you could have groups 1 through 10 on 100 data pieces, and then figure out exactly how you can figure out exactly what the difference is between all of them. So given that we know there is a significance in the means, one way ANOVA will not tell us where the significance occurs in a group of k groups. In order to do this part, you want to do something called a Tukey test or a Sheffe test. Honestly, I don't know how to pronounce this, so just, say this however you want in class. Tukey is for equal sample sizes and Sheffe is for unequal sample sizes. The Sheffe one is a bit easier for me to comprehend, so that's the one I'm going to follow. First, you're going to have your critical value. That is the value that you want to exceed in your f-test, and that is calculated by getting the f critical value given your degrees of freedom for 1 and 2 and your alpha, and then you multiply it by k minus 1. So that will give you the ratio that you're trying to hit. And the F statistic is calculated by taking the two separate means that you care about and squaring them, and then dividing that by the mean squared error, which is also multiplied by the number of samples in the first plus the inverse of the number of samples of the second one. In these cases, the Tukey test and Cephei test should be used rather than using multiple t-tests. The multiple t-tests do not account for the multiple comparisons that you're making and can lead to more false positives than something like the Tukey or Cephei test. So moving on to multi-factor ANOVA. First, we should discuss what multiple factors are. Many factors are typically taken into account when you're trying to explain something or trying to predict something. In GLM, we had multiple predictor variables for one response variable. With ANOVA, we can skip multiple two-group comparisons in t-test and use the multiple factors as a way to get around that. It allows you to more efficiently test large amounts of factors, and due to the design of ANOVA, you actually require fewer observations to get the same statistical significance or p-value that you would need from the t-test. You can also control for multiple factors as well in ANOVA and basically test factors while controlling them for given conditions that we'll discuss later. When you have multiple factors or multiple predictors, whatever you want to call them, or independent variables, you can start getting interaction effects. So rather than just looking at one effect, like in the graph to the right, like challenging test versus an easy test, or achievers versus slackers, you can combine the two in plots and see how they result. In this case, if you look at the mean of the challenging test collapse across the different types of people versus the mean of the easy test collapse across the difference, you'll notice that there is actually no difference. Same thing happens when you take the mean of achievers collapse against the type of test and slackers. So in this case, the two main effects of the type of test and the type of achiever or slacker doesn't have a significant effect. But when they're combined, then we can see that slackers try harder on easy tests while achievers try harder on challenging tests. So basically points 1 and 2 are the main effect, and 3 is a two-way interaction of those two effects. So what we will be interested in is which of these effects is significant and if the interaction is significant. This same idea can actually be applied to three-way interactions or n-way interactions. You can have as many as you want. The only thing is they start getting much more complicated at that point. So here we have the same data set, except in this case there's also the factor of females versus males. It becomes much more difficult to display multi-factors after the third one. It includes a lot more plots and it becomes a bit difficult to talk about. In these cases, for any type of interaction, a good way to talk about it is to say that the main effects, in this case, let's say the main effect for the test is modified or qualified by the personality. That is referred to as a two-way interaction, and we see that happening both females and males. But on top of that, we see that there is a difference here for the females and males in that they switch on their two-way interaction. In that sense, we can say that there's a two-way interaction between test and personality that is qualified or modified by the gender. So that would be the three-way interaction. And using this pattern, we can actually express any sized effect this way, using the modified or qualified terms. So let's look at an example here. We have a 2x2 table, which is two groups of two factors each. The within group error is actually going to be lower when you include the gender compared to excluding the gender. And so I calculated that for us here. So if we take all the experimental group 1 and calculate their group sum of squares, we get 10 for each. Added together, we get an SS of 20. But if we look at the individual groups where we take the experimental group by the type of gender, then we would have 2 plus 2 plus 2 plus 2, which is 8. So controlling for the error variance actually helps when you add in these new factors, and that allows you to increase the power of your test by doing this. So this is just me taking some required sums. Basically, the best way to do a two-way interaction is to first get all the sums for the individual cells and then calculate the aggregate sums below that. So group 1, the sum between on all male and female is 18. Group 2, it's 18 plus 24, which is 42. The links down here go through all the math that is required to do this by hand. And I think it's of interest, and you should have a pretty good sense of how this occurs. Given that we get those sum of squares, using the formulas in the linked websites, you get this table. So here what we see is that we have a significant main effect of the experimental group and also of gender. But when we include gender and an experiment, we get a probability of 1, which is basically saying that there is no variance left that can be attributed to the interaction of gender with experiment. And the website link below allows you to put in this data set yourself and can give you a chance to try out two-way ANOVA yourself. Moving on to complex designs. What's nice about ANOVA is that it's robust and you can actually do a lot of interesting tests with it. I will not go into detail about all of them, but I will cover some here right now. So first is we can do between group and repeated measures designs with ANOVA. So we can remember that t-tests for dependent samples exist. That's basically when you take the difference between two equally sized samples, usually when you're dealing with pre and post test. ANOVA needs to know whether you're doing a repeated measure or one of these dependent sample tests, because the math required for it is slightly different and just different enough to change the answers. If you are dealing with repeated measures or dependent samples, you know, pre and post test or pre, middle, and post test or whatever, then you have a repeated measures factor or a within subjects factor. So the computations, as I said, are different, but the interpretations of the results are going to stay the same. And then if you compare across different groups of subjects, so like males and females, group 1, group 2, that's going to just be between groups factor, and that's the examples we already covered. Now one thing you can do is have a within designed ANOVA. So in this case you have between group factor with a within subjects factor. So maybe males and females, so that's one between group factor, 2 levels, and each takes a test beginning and end of the semester, which is a 1 within subject factor of 2 levels. The resulting table will look like the one that we already seen. It will just require different calculations for all of the numbers. There's also something called incomplete or nested designs. This is basically what happens when you have too many factors to properly test everything. So let's assume a test where we want to have 4 different additives in a car, and we're going to try that against 4 different cars with 4 different drivers. To figure out how many conditions you would need to test all of these, you just multiply the number of conditions for each of the factors. So this is 4 times 4 times 4, which ends up being 64 individual groups that you would have to probably get at least 3 or more observations from. It's simply not feasible to do this, so sometimes we can use something called Latin squares, which is demonstrated on the right. Basically, each driver and each car experiences each of the conditions once. The mixed model ANOVA or ANCOVA can work with this and is robust enough that it can handle this stuff without you worrying too much about compromising your ability to make any conclusions from the tests. Now, moving on to analysis of a covariance or ANCOVA. So the general idea between analysis of covariance is that you want to try to control for some of the factors in your ANOVA. We know that if you add in additional factors, you can reduce the error sum squared, which in turn increases the power or sensitivity of your design. This can be used on continuous variables as opposed to simple counts, so stuff like a factor that is just literally counting the number of students isn't as interesting as, let's say, some continuous variable like a ratio or correlation. When you include these factors in your design for control purposes, then they're called covariates. So, fixed covariate. First example that we can think of is, let's say we have a test where we want to see how math skill changes given a student being randomly assigned to one or of two textbooks. So he either gets textbook A or textbook B. Now we know that the IQ for a student is going to be tightly correlated with the amount of math skills they have. So given that we know this, we can actually include IQ in our experimental design in order to eliminate the differences that we see in math skills between the two groups that can be accounted for by the IQ. So what you would do is in each group you would compute the correlation coefficient between IQ and math skills. And so from this given R, we can estimate the amount of math skills that are accounted for by IQ, and the residual or the variance is not explained by just IQ. And this residual variance can be used as the estimator for the true error of the sum squared. If there's a large correlation like this, then the reduction in error sum squared can be very large and can be increasing the power of the test significantly, which allows us to not have to get as many subjects or observations taken. As the error of the sum squared decreases, so does the mean squared error as well. Because of this, the F ratio, which is the mean squared effect divided by mean squared error, in that case the F ratio is going to be increasing in size. You can use multiple covariates, so maybe something like IQ and motivation, spatial reasoning are all important in determining math skills, and what we really want to know is, does math skills improve due to the fact of just the test alone and not these other factors? In those cases you would need multiple correlation coefficients, but there is chances where the F gets smaller. So if the covariate that you include is not actually correlated with between group factors, then what ends up happening is that the covariate partitions the main effect SS from the between group. Basically it's taking away from the numerator portion of the F, which means that you end up losing power in your experiment. So by trying to eliminate an effect, it's actually possible that we could actually hide the true effect. So maybe as IQ increases as well as math skills when you're using those books. If that happens, then you can't actually see the difference in the math skill improvement only because you're including the IQ. Extending the fixed covariate some more, if the covariate is affected by between groups factors, so in the case of the IQ increasing as well as the math skills, then it's appropriate to compute the adjusted means. In this case, the means that you get are after you removed all differences accounted for by that covariate. And so in this case, when the ANCOVA assumption is violated, that that the covariate does not change across between group factors, then you can use this estimation to get an estimated of significant interaction between the factors and covariates. The thing is, ANCOVA is robust enough that it does this regardless, and it's not too much of a concern for most tests. In ANCOVA, we're assuming that the covariate is going to be fixed and isn't supposed to change. But sometimes we actually care about how the covariate changes along with the dependent variable or the response change. This is typically measured at each time point that the dependent is computed, and basically you can compute the correlation on those changes in a covariate compared to the changes in the dependent variable to get a sense of how covariate is actually changing across these experimental conditions. Moving on to multivariates, I'm going to be talking about MANOVA, but MANCOVA is basically the same as MANOVA, it's just that it has the included covariates that we just discussed. So as a reminder, multivariate refers to multiple dependent variables, so multiple response variables or Y's in your GLM. Multifactor refers to multiple independent variables, so that's when you have multiple predictors or X's from GLM. So first let's look at between groups design for MANOVA. In this case, you have different textbooks and you see if math and physics skills are going to improve given one of the two textbooks. MANCOVA can test this, and math is largely the same as before. In this case, though, you have the F statistic called the multivariate F-stat, which is also called Wilks lambda. Here it compares the error variance over the covariance matrix and the effect variance over the covariance matrix. This covariance is actually based on just dependence and how they change. There's a good chance that they're going to be probably related, so it's a good idea to have this. Correlated measures, when we know that there's a correlation calculated, actually let us learn something new. If we didn't do this, if let's say we just did two ANOVAs, if they were correlated, then you will basically be doing the same measure twice, and you introduce the potential for false positives and other issues. Going along with between groups design, if that overall multivariate test is significant, you can conclude that the respective effects, textbooks from before, is significant. Now, the next question is, which of these dependents changed? And in this case, you would use a univariate F-test on each of the dependents, or just the ANOVA for those. And then you can, that will allow you to interpret which ones actually changed. The thing is, though, you can't do this latter ANOVA univariate F-test before you've done your MANCOVA test, because you need to first know that there is a significant difference between them before you try to figure out which one it is. Otherwise, you are introducing the potential for false positives. MANCOVA can also be used for repeated measures design. So for example, if you have math and physics skills that you're tracking when you are looking at two different books at the beginning and end of the semester. The logic of the significance testing is the same again, but the math is going to be slightly different. MANCOVA can actually be used for the significance test of univariate repeated measures factors, and this is typically done when you have more than two factors. So basically, if you have a lot of time series data points that you're taking, MANCOVA will be actually pretty good and robust enough for this stuff. Now moving on to something called contrast analysis and post hoc analysis of your ANOVA tests. So when you have an experiment that you want to make a hypothesis on, let's say that your hypothesis is going to be on interaction, it's simple enough to make that that test. You just say, you know, that your hypothesis is that effectiveness of a textbook on math skills is qualified by a student's gender. You do the same stuff as before and test, and you can say whether that's true or not. If you actually want to say something more specific, like maybe the effectiveness of a textbook on math skills is modified by gender on one book but not on the other book, then at that point you need contrast analysis to get this type of complex effect. In contrast analysis, you basically test the statistical significance of predicted specific differences in particular parts of complex design. It's a long statement. I could not figure out a more concise way to explain it, but in our example it would be basically saying that gender plays a role in one book but not in the other in improving math skills. Any complex ANOVA design will typically have some sort of hypothesis on this, so you can actually test these things. Now, post hoc comparisons. These basically happen after the fact. So let's give an example to show how you can fall into some pitfalls with this. Let's say you write down a random number from 1 to 10 on a piece of paper, and you do this 100 times, and you put it in a hat. Now you draw 20 samples of 5 numbers each, and you compute the means for each of these 20 samples. Overall, the average is going to be around 5, but in any given 5 of them, it might be significantly different from that. So what are the odds that 2 of these are going to be significantly different from each other? Well, it's actually going to be a pretty good chance because you have 20 samples, and if you have especially a confidence value of 0.05, then there's a good chance that this could happen. So basically what's happening is that you're taking 2 extreme means and comparing them, and that's not the same as taking 2 samples and saying that 2 samples are going to be statistically significant compared to of all means that any 2 will be significant. So to correct for this, you do post hoc comparisons, and basically you take in post hoc analysis, you take the most different means from the k total amount, and then you try to correct for the advantage that you get from choosing how separable those means are. Many post hoc tests exist. It's going to be up to you to find which one is most appropriate for your particular case. So now moving on to what happens when you violate any of the assumptions that ANOVA is running on and how to deal with that. First, there's the distribution assumption, in particular that the response or dependent variable is going to be normally distributed. And of course, it's going to be at least on the interval scale, so something like Fahrenheit or Celsius, but it doesn't need an absolute zero like Kelvin. So it should be normally distributed though. If you violate that, if you have data collected that is not normally distributed, if the kurtosis is greater than the normal distribution, so it's very peaky, then the F is going to be usually too small and you can't reject a null hypothesis. If the kurtosis is too flat, then the opposite happens and you have a higher chance of an actual false positive. The skewness of the distribution doesn't really have a big effect on this. F-test is robust enough that it doesn't care. And in order to get over these hurdles, it's typically better to have larger number of observations for any one cell of interaction to keep this from being an issue. So in the two-way ANOVA group, you have experiment group 1 and males, experiment group 1 and females, experiment group 2 and males, experiment group 2 and females. In those cases, you want to pump up the numbers for each of those combinations. If you do this, that can actually, the higher amount of observations will actually help remove the effect of the non-normal dependent distributions thanks to the central limit theorem. Another assumption is that you're going to have a homogeneous variance across the different cells. So the error sum squared requires that you're adding up all the sum squared from each cell. In that case, as we had before, it was the same value across, 2, 2, 2, 2. So what happens when you actually have maybe 2, 4, 10, and 2? In this case, it's actually harder to make an argument for what the within group variance is when the variance is changing. In this case, though, luckily the F statistic doesn't actually get impacted too much, and it's able to handle the fact that that changes so much. The only issue occurs, though, is if the cell means and the cell variances are correlated. So given those cell interactions, so group 1 and males and their mean and variance compared to the group 2 and males and their variance and all the other associated cells, if you plot the variances for those versus the means for those subgroups, if there's a correlation, then you have an issue, and that's typically dealing with outliers in the data set, and your F statistic is going to be skewed, and you're going to have to correct for that in some way. The next thing is that you also want homogeneous variance and covariance, in particular in multivariate dependent designs. Basically, the covariances need to be homogeneous across the cells. They shouldn't be changing. And the variance of the individual dependents also needs to be consistent. So the Wilks lambda that's used in MANCOVA, we don't actually know enough about it in order to have a proper understanding of what happens when you break these violations. So you need to be really careful, and any univariate tests that you do afterwards to figure out where the differences lie need to be scrutinized even more. In ANCOVA, the covariation changing across cells leads to serious biases, but there are specific tests that are designed to see if that assumption is being violated. So the last assumptions that we're dealing with is for sphericity and compound symmetry. So in repeated measures, usually it's best to rely on MANCOVA instead of repeated measures ANOVA. ANOVA requires two things to work: that the covariances across the subjects, so the repeats, and the variances of within group effects across subjects stay the same. So it needs compound symmetry. So that within group effects we already talked about, but basically the covariance cross the variance and the covariance as you move through different tests. So going from, let's say, pre-semester, mid-semester, final semester, those variances need to stay the same. And on top of that, you need to assume that the pre and post tests are uncorrelated from each other. If they're correlated, then you have biases, and this refers to sphericity. The second one is necessary for ANOVA to work, so you definitely need to make sure that they're not correlated when you're doing ANOVA. But the first, you just if you have it, then that's good, but it's not necessary exactly. MANCOVA is actually able to deal with these violations of sphericity and compound symmetry. Prior to MANCOVA, when people were just using ANOVA, they had a lot of issues with this, and so they had to create a lot of compensations for this stuff. But in your case, you should typically be using MANCOVA if you have a concern for this. Sometimes it is a bad idea to use MANCOVA, and that's usually when you have few subjects and many levels. So let's say you have 12 subjects and you have 4 repeated measures like beginning of the week, middle 2 days of the week, and end of the week, and you have 3 levels like trying out 3 different formulas. This 4-way interaction would need k-1 to the p power of degrees of freedom. In this case, it's going to be 16. With only 12 subjects, you can't actually perform this test. So you actually have to rely on ANOVA in this case. There will be differences between MANCOVA and ANOVA, which usually indicates that there is covariation of the repeated measure. Okay, as a summary, ANOVA is used for comparing multiple means simultaneously, typically when you have more than 2 groups, because at 2, the t-test can do just fine. ANOVA can deal with multiple factors, so let's say gender and test and other ones. You can go as complicated as you want. It can also deal with repeated measures as well, though it's probably best only when you're dealing with 2 repeated measures as opposed to multiple ones. At that point, MANCOVA takes over. So MANCOVA deals with multiple dependents. It's the best approach when you're dealing with repeated measures. ANCOVA is used to control for any factors that you want to tweeze out of the test. MANCOVA is the same as MANCOVA, just now you can also control for factors with multiple dependents. And then on top of that, the underlying distribution of the dependent variable responses doesn't really affect the significant test much, but the changing variances and covariances can affect the interpretation and should be watched out for. Here is the research I used to create most of these slides. If you have any more detailed questions, this link might be able to answer it. Thank you for listening to me.